Bismillahirrahmanirrahim. Dear students, I am Adnan and this is Biology online class for grade 9 seniors. Dear students, you almost covered 5 chapters in your grade 8. So we will start from chapter 6 and the name of this chapter is enzymes. Dear students, the life of living organisms is the reflection what is going on inside their bodies. Dear students, metabolism is the set of biochemical reactions in a living body. The word metabolism is a Greek word which means change. Ibn Nafis was the first person who gives the concept of metabolism. He stated that there is a continuous change in living organism's body with the passage of time. Dear students, metabolism is the set of biochemical reactions in a living body. There are two basic types of metabolism, and these types are anabolism and catabolism. The anabolism is the type of metabolism which is called to be a constructive process in which synthesis of new substances takes place, and catabolism is a destructive process Dear students, the anabolism example is photosynthesis. Photosynthesis. What is photosynthesis? Photosynthesis is the process by which plants prepare their food in the presence of sunlight with the help of chlorophyll. So this is a constructive process. So photosynthesis is the example of anabolism. And the example of catabolism is respiration. Respiration is a process by which food molecules are broken down and energy is released. Once again I am repeating. Respiration is a process in which food molecules, especially glucose, is oxidized and energy is released. So this is a destructive process by which we achieve the energy, which is very important for us, which is very important for every biological process which is going on in our bodies, and which is going on in every living organism's body. So once again I am repeating, metabolism is the set of biochemical reactions in a living body. It has two types, anabolism, catabolism. Anabolism is a constructive process in which synthesis of the new molecules take place. Photosynthesis is a big example. And catabolism is a destructive process, and respiration is a big example of the catabolism. Now, the question is how these reactions take place. They need any facilitator? So my answer will be yes. They need a facilitator. they need a chemical substance which catalyze, which speed up these chemical reactions which are going on in photosynthesis, which are going on in respiration. And these catalysts are called biocatalysts, as they are speeding biochemical reactions. And these biocatalysts are called enzymes. So our topic is enzymes, and we will talk about enzymes in this chapter. We will discuss the structure of the enzyme. We will discuss the characteristics of enzymes. We will discuss the different factors which are affecting the rate of the enzymatic activity. And dear students, we will also discuss the modes of actions of enzymatic actions, which are two types, and inshallah we will discuss it in detail in our coming lecture. And specificity of enzymes. So these all about, we will talk in this chapter, we will talk about the enzymes, different aspects of enzymes. Hope that you have learned the definition of the metabolism. You learn what is metabolism, what are its key types, and what's going on. Now I'm going ahead and I'm starting about enzymes. So first of all, I would like to define definition of enzyme. So enzymes, the catalysts which speed up a biochemical reaction. Enzymes are biological catalysts. These are biocatalysts which speed up a chemical reaction. But you have to know the definition of catalyst too. Catalysts are chemical substances which speed up any chemical reaction, but itself unused or unchanged. In that way, enzymes are biological catalysts, so they speed up of chemical reaction, but itself unchanged or remains unchanged or unused. They are the facilitator which speed up a chemical reaction. So the first question which arises in the mind of a student, that why we need enzymes in a biochemical reaction? Whether we are not able to perform any chemical reaction without enzyme. So for this purpose, let me to give you a comparison of two reactions without a catalyst and with a catalyst. This is the reactants. Reactor. This is the product. All of you know that in a chemical reaction there are two types of substances. Before reaction there are called reactants, and after the reaction we got product. So if I want to start a chemical reaction, I need activation energy. This is the amount of activation energy. Let me define activation energy also. Activation energy is the minimum amount of energy required to start a chemical reaction. So, if I want to start this reaction, I need that amount of activation energy. But if I put a catalyst, then I need that amount of energy. It means that if I am doing a chemical reaction without a catalyst, it needs that amount of activation energy, and with the help of catalyst or enzyme, I need that much energy. It means the amount of activation energy in the presence of a biocatalyst is reduced. And that is the key point. That is the key point, that actually enzymes speed up the chemical reaction and it reduces the activation energy of a biochemical reaction in a living body. This is the key point, that enzymes reduce the activation energy. The activation energy which is required to start a chemical reaction, to start a biochemical reaction in a living body. It reduces the activation energy. Dear students, hope that you learn about the activation energy, and you learn that if we perform a chemical reaction without a catalyst, without an enzyme, how much activation energy need, and with the enzyme involvement, how much activation energy it needs. So definitely the activation energy is reduced. Now the second point in the mind of a student that arises is how enzyme is converting, what enzyme is doing with the substrate. What is substrate? Substrate is the substance or the chemical on which enzyme is acting, and product is substance which we obtain after a reaction. So what enzyme is doing with the substrate? This is a question in every student's mind that what enzyme is doing with the substrate. In some cases, it is bringing the substrate molecules in a precise order. In a precise order. Secondly, it is disrupting the distribution of charges in the substrate. And in some cases, it is altering the shape of substrate. So there are too many possibilities and even we have experimental proofs also that how enzyme is, what enzyme is doing with the substrate. But few of them I have mentioned here. Hope that you have learned what I told you about how enzyme is doing, how enzyme is acting on the substrate. Dear students, after that I am going to start the characteristics of enzymes. The characteristics, there are different characteristics of enzymes. First is all enzymes are proteins. All enzymes are protein in nature. Enzymes are protein in nature. But I will add here that not all enzymes are protein. Very few, like ribozymes. Ribozymes. They are RNA enzymes. So majority of the enzymes are protein in nature, and these proteins are synthesized by our cells. These are synthesized by our cells. They are produced inside our cell by a process called protein synthesis. which is also called to be central dogma of molecular genetics, which is also called as a gene expression. In our DNA, there are genes, and these genes actually give information in the form of messenger RNA, and messenger RNA then to ribosomes, and on the ribosome, by the help of transfer RNA, they are preparing special sequence protein, which then converted into different enzymes in endoplasmic reticulum and then in Golgi bodies they are stored. All of you are very well aware about the ribosomes. You have studied the structure and function of the ribosome in chapter 4, and all of you know that these are called the factories of protein where proteins are synthesized. So actually proteins are synthesized, but these proteins are then used as enzyme. So our first characteristic, that all enzymes are proteins. They are protein in nature, except I have written the example of ribozyme. These are RNA enzymes. These are RNA which act as an enzyme, and then they are called ribozymes. Second, I have given you a complete description that how these proteins are synthesized inside the cell. So the second characteristic is the rate of enzymatic reaction. Second characteristic is the rate of enzyme activity. So dear students, this point is already clear that I told you that enzymes reduce the activation energy, they low down the activation energy, and with the help of the enzymatic activity are enzyme mediated reactions speed up chemical reaction millions of times. Means that the reactions in which enzymes are used are millions times faster than those chemical reactions which take place without enzyme. Now the third one is the enzyme specificity. Third one is the enzyme specificity. Enzymes are very specific in their function. Enzymes are very specific in their function. I will mention here two examples. Pepsin is an enzyme that usually present inside your stomach. They digest, they enhance the digestion of protein. All of you know. And amylase is the enzyme in the small intestine. Actually, amylase is secreted by the pancreas, but it acts in the it performs its function in the small intestine. It digests the starch. If I put pepsin in the starch, if I use the pepsin for the starch digestion, it will not work. If I use amylase for the digestion of proteins, it will not work. There are many enzymes like DNA polymerase is the enzyme, or the group of enzymes, DNA polymerase 1, 2, 3. These enzymes are usually used in the DNA replication. RNA polymerase enzymes are used in the gene expression. Usually in the gene expression or in protein synthesis, the RNA polymerase enzymes are used. There are many, and about 2000 types of enzymes are present inside a cell. Every enzyme has its specific substrate, a specific substance on which it has to work. It means that it's not true that if we say that one enzyme is catalyzing many chemical reactions, different type of chemical reaction, not different type of chemical reaction. It will work on its specific substrate. I think it's clear. Now the next point, or the next characteristic is the shape of the enzyme. Shape of enzyme. When I was talking about its catalytic activity, in a student mind, this question should be arise that what should be the probable shape of the enzyme, how it's reacting, how it's engaging the substrate. So, dear students, there is a specific site in an enzyme, if I call it as a enzyme. This site is called active site. It can only engage and attach substrate with the active site. It can engage and attach the substrate only with the active site. So the active site is the site by which it can attach, by which substrate can attach with the enzyme. The rest of the enzyme is the non-catalytic part. Only the catalytic part is the active site. And secondly, they are made up of many linear chains of amino acids. But these linear chains fold in a way that it is converted into a globule shape, a globule. It is converted into a globule, and it gives a globular shape. There are beta sheets, many linear amino acids, chain of amino acids mean that many amino acids attach with each other and forming a chain. And these linear chains then fold and give a globule shape, a three-dimensional shape. And this three-dimensional shape is then called as an enzyme. And the only catalytic, its catalytic part is called as active site. Now that we are moving ahead, and let me to tell you about activators and inhibitors. Activators and inhibitors. Activators are the chemical substances which activate the enzyme. Usually substrate is called as the activator, because if substrate is present, then enzyme should be activated and the genes should be switched on to make a particular enzyme to catalyze that substrate. So usually the substrates are the activators. Now the inhibitors. The inhibitors are the chemical substances which stop the chemical substances which stop enzyme activity. Inhibitors are the substances which stop the enzymatic activity. There are many inhibitors, reversible inhibitors, irreversible inhibitors. One of inhibitor I want to mention here is cyanide, which is thought to be a very harmful poison. Cyanide is an inhibitor, and it is an irreversible inhibitor, and usually it arrests the active sites. All of the inhibitors, how you can stop an enzyme from its working? Very simple. You can block its active site. If you block its active site, an enzyme will not work. So basically, the cyanide are the different inhibitors usually block the active site of the enzymes, and about the cyanide, it is clear that it usually blocks the active site of those enzymes which are working in the cellular respiration process. So, dear students, inhibitors usually block the active site of the enzyme, and thus the enzyme activity is ceased or stopped. But there are some reversible inhibitors also. Even an enzyme itself can inhibit itself. For example, if there is no substrate, then we have to switch off the production of enzyme too. So enzyme itself, if there is no substrate, it can act as an inhibitor. Now moving ahead, let's discuss the very important property, characteristic of enzyme, that is the cofactor. Dear students, the non-protein part of the enzyme is called cofactor. You should think, dear students, you should think that I always say that enzymes are protein in nature and all of the things are doing by the enzyme, and enzyme is catalyzing the biochemical reactions. So, a non-protein part should be the part of an enzyme. Some enzymes, dear students, some enzymes. This, the non-protein part, this is called the cofactor. This is active site, and this is the cofactor or the non-protein part of the enzyme. Dear students, without cofactor, this enzyme is called apoenzyme. And with cofactor, then it is called holoenzyme. Apoenzyme and holoenzyme.
Synthesis.